All right, thank you so much for joining us this morning. I guess we better get started with our event today. Let me begin by doing a quick land acknowledgement. Let me begin by acknowledging that we are privileged to gather today on the unceded traditional territories of the Squamish, Tsleil-Waututh, Coquitlam, and Musqueam peoples. We thank them for having cared for these lands and waters since time out of mind, and look forward to working with them in partnership as we continue to build this great city together. If you know whose territory you're currently on, please take a moment to reflect on this. And if you don't know whose land you're on, I encourage you to look it up and make yourself familiar with the Indigenous peoples whose land you occupy. Good morning and welcome everyone. My name is Cynthia Henson and I work with the Simon Fraser University Faculty of Science, and I look after their outreach and engagement program. And I will be your host for this morning. This is Science Rendezvous. This happens every year. It's actually a science fair that's celebrated nationwide once a year. And we've been doing this for over a decade now. And what this is really is we bring together scientists from different host institutions across Canada, and we celebrate science contributions and learning together. And Simon Fraser University has been one of the biggest stagers of Science Rendezvous here in British Columbia, and we're happy to have you with us today. So for this year's theme, we do follow the astronomy theme, and we do this also because we had always wanted to celebrate International Astronomy Day. So for this morning's session, we are happy to feature our friends from School District 43 in Coquitlam, because Simon Fraser University Faculty of Science has partnered with this School District 43 in a collaborative event called Student Spaceflight Experiments Program. We've been doing this with this school district for 2 years now, and we are always happy to have our faculty members and graduate students mentor the student teams from this school district, all the way from middle school and high school. And we're always excited to see their experiment proposals each round. So with me on the call today are friends from School District 43, Stephen Whiffen, who is their Director of Instruction and Chief Information Officer. We also have Nicole Cruz, one of their school district teachers who've been a great help in helping our student teams with their presentations this morning. And we have Marisa and Selina from Hillcrest Middle School. We have Jisong Han from Charles Best Secondary, and we have J.E. Lee and Yong Lee from Port Moody Secondary, all joining us this morning. Thank you so much. So at this point, I'd like to turn over the floor to Stephen Whiffen, SD43 Coquitlam's Director of Instruction and Chief Information Officer, to tell us a bit more about this Student Spaceflight Experiments Program. Over to you, Stephen.
Thank you very much, Cynthia. Really appreciate it. And I want to just start out by saying how important it is for us in the Coquitlam School District 43 to have this working relationship around STEAM and science with Simon Fraser University. I think one thing that's really important to point out at first is that STEAM is an official district goal in School District 43. We've identified it as a priority area development, and we've done so because we believe it's incredibly important that every child has an opportunity to be exposed to STEAM ideas, how STEAM affects the world around them, and also STEAM ways of learning. And if you don't know what STEAM is, it's science, technology, engineering, arts, and math. So those pieces individually are all things that we're all familiar with. As you know, when we put them in the context of a school setting, you probably think of them at first as subject areas. But actually, what we're trying to do in K-12 education is start to think of them as something that's more integrated, something that's more tied together. And when we do that, there's a couple of things that emerge. So one is that we engage in all of those disciplines in ways that interact with each other. So when we're doing science with technology, and we're engineering solutions, and we have to apply math in order to do that. and increasingly the ideas that relate to art is becoming part of it, which is why we refer to it, we include the A and refer to it as STEAM. And you're going to see all those elements in this presentation today. And I think that's something that I wanted to sort of set off, so that when you're looking at the work that these students have done, this amazing work, you're making sense of why this is a priority for us, and how the STEAM fits into all of this as we go. Really need to emphasize that the reason that we have put so much priority on STEAM is because we think that it's an equity issue. We feel that it's critically important that every single child, regardless of their background, regardless of the school they go to, has a chance to engage in these ideas, and maybe even more importantly, this way of learning. And that's because the entire world around them is evolving so quickly and has so much science and technology embedded in it, that it's critical that they understand that world and how those ideas and those ways of the world working develop, in order for them to interact with the world more safely and more effectively, and to give themselves the most opportunity possible. That doesn't mean every single student is going to go into STEAM-related careers. Some will, and that's important, because we need that next generation of leaders who are developing the amazing things that are around us, who are developing the next smartphone, who are developing this Zoom technology that we're working on today, who are developing the next medical breakthroughs. All of those things are going to be dependent on STEAM-based engagement in some way. But even if you're not going into STEAM as a career, it's still valuable, and actually I would argue critical, for every student to be exposed to it, because the world that they're interacting with every day, just like you and I are interacting right now, is driven by those tools and those technologies and that science. And if you don't understand it at all, then you're just a visitor to that world, and it has a certain amount of control over you, because you just do what it has you do. We want citizens who understand that world that they're engaging in. And even if it's not for career reasons, even if it's just because you're a citizen of that world, to know how the world works, to know some of the pros and cons of that world, to know what the opportunities are in that world. We think that's something that is necessary for everybody. And so if every student isn't getting exposed to that knowledge and those ways of learning, then we think it would be putting some students at a disadvantage. So it's a very, very critical initiative for us. And with that in mind, it's also complex. It's very, very sophisticated ideas that are not always really accessible to the typical K-12 student. So we need to look for ways to have opportunities for kids to engage it in meaningful ways. And one of the ways that we think is really important in doing that is the project that we're talking about today, the Student Spaceflight Experiments Program, or what we call SSEP for short. The other way that we think is really critical to do that is community engagement. And our work with SFU, I don't think any of this would be possible if we didn't have the connections and mentorship provided by students at SFU who are working with our students that help middle and high school students to unpack really complex ideas and engage with them in a way that relates to their individual goals and ideas that they come up with. And we can't do that in every classroom without having a little bit of community engagement. And so that relationship with SFU is really important. But then you ask, well, okay, so this SFU thing or this SSEP thing is important, but what's it really about? Okay, I see this about students. I see this about spaceflight in some way. That sounds interesting. It's something about experiments, and it's a program. Well, the place to start is the image that I have in front of you right now. That is, if you don't know already, the International Space Station. And that is what this is all about. This is an opportunity. SSEP is an opportunity for one group of students to have an experiment that they design that makes sense to do in space, to actually go into space, to go to the International Space Station, where an astronaut will actually do that experiment in space, collect results, and bring all of that back to Earth. And when it comes back to Earth, that student team gets to analyze the results of that experiment and compare it to how the experiment went here on Earth. And in doing so, develop some insight into not only space, but hopefully our world and the way our world works as well. And I can't think of anything that is more engaging, that would motivate students more than the opportunity to have their experiment go and have this actually be done in space, have those results come back, and have an opportunity to actually change the world by informing it with new information. That is authentic science. And so when we start to break that down a little bit, you kind of get to a summary of what this is all about. This is all about authentic science. We want students to actually do real science. And we know that the learning will be better when the science they do is real. And that's part of the goal we have for STEAM education in SD43. And I know that's part of the goal that SFU has for students as well. And so that's why we're so committed to working together on that. Now, the unfortunate part is only one student project gets to go into space. And when you think about it, you can imagine why that's the case. It's a very, very expensive proposition for an experiment to go into space. And while we wish every student would be able to have that opportunity, it's just not possible. So what SSEP does is it recognizes that reality, that constraint, and creates a bit of a structure that ensures that hundreds of students, thousands of students are able to engage in that same authentic science experiment and experience, and then recognizing that at the end, unfortunately, only one can get picked to go up. But the process in getting there, the process in trying to be that one that gets to go up, is in and of itself engaging in this authentic science. And that's so important to everybody. So how does this work? The next slide, Nicole. So the way it works is it's actually a pretty big undertaking on the part of the district and our partners. This is a supporting webpage that we have here, and it has a bit of information that I can break down for you to help you make sense of it a little. One is it requires a bit of funding in order to do it, and the funding comes from a couple of different places. It comes from the National Center for Earth and Space Science Education in the United States, which makes the project possible through grant and funding programs that they have to facilitate this for schools. But more locally, it comes from local relationships. And you see on this page here, one important one is the School District 43 Education Foundation, who contributed approximately $30,000 towards the initiative to make it possible. And then, of course, our relationship with SFU, which is equally necessary. Without these connections and contributions, we wouldn't be able to pull off a project to this scale. But how it works from here is it's a multi-month exercise. So what you see on the screen here was originally, it came alive in August of 2021. That's effectively when the project kicked off. When students came back to school this year in September, we had already prepped teachers to engage students with this project. And that was very necessary, because one of the real constraints of SSCP is it is a really crazy timeline. There's a lot of work to be done. And the student teams that you see here today, when you see their amazing work, you're going to realize they had to do an entire authentic science cycle in less than 3 months and get to the place where they had a real proposal that would represent the type of proposal a scientist would do. And the write-up and detail that goes into that, interacting with real scientists to be sure that the science that was in it was correct and that they weren't making assumptions or errors in that. That interaction takes time. And it's a real testament to these students here today that they went through the intensity of that exercise over about a 3-month period to get to the point where they had developed really, really high level, amazing projects. And we're going to get a chance to look at some of those next. But to make sense of that and to add some authenticity to it, You also have to recognize that actually there was hundreds of students from School District 43 who worked on projects. And that's really important because, again, I said earlier, this is an equity issue. This is about making STEAM experiences available to everybody. So, of course, the exercise is open up to everybody. And we, through the website that you see here, provided resources for teachers and student groups to use to help them participate. So if Nicole, you can just move to the next slide. Now, in order for it to be authentic and in order for this type of structure to work, it takes a lot of people working. And it takes a community, actually, is how I'd frame it. And we've already talked about the role of the district and the role of the university, Simon Fraser, being part of this. But actually, the community needs to be even broader than that. Because we had hundreds of students create proposals, there was a highly engaged structure in determining which proposals were the best candidates to ultimately end up being one that goes into space. And we had an amazing commitment from a group of people to make sure that that was a really genuine, authentic experience, a judgment experience for all students who participated. I think that's critical because if you, as a student, have taken the time to participate in this and have done your best to put forward a proposal that you hope could be considered to go into space, you may not get chosen, but it deserves to be looked at by real scientists and others who are actively engaged with the work you've done and give it the attention it deserves to determine whether or not it meets the bar for being the one that's chosen. And we were very fortunate to put together a fantastic team to support with that. You actually see them here. You can see at the time, some of us were in the live event and some of us were on our Surface Hub in a Zoom call in behind. There was 4 other people in behind there, hybrid meeting. And we met for an entire day reviewing projects. And the way it worked is schools reviewed projects that were submitted at the school level to determine the best projects at each school. Those projects were then forwarded to this team that you see here in this photograph. And this team reviewed 30 projects, the kind of 30 finalists. And so if you can just flip slides, Nicole. You can see the type of people we have involved. I'm not going to read the list. The group that you see on the left is a school district staff ranging from assistant superintendents and myself to our STEAM coordinator, our numeracy coordinator, and Nicole, who's on the call here, such an important part of the process because she facilitates so much of it. But on the right side, you see people from outside the school district. You see Eileen Vanderfleur-Keller, who's a teaching professor at SFU in Earth Science. Andrew Blabber, who himself is sending actual science experiments up into space on a regular basis. These are people who are highly in tune with the science world in this area. And it's amazing for us to be able to get our students access to people of this caliber. And these are the people that were judging their projects to make sure that every one of them was getting the due attention that it deserved. But we also had people from industry. We had Brad Klink, who's from IBM Canada, again, part of the technology industry, very important for that connection. And as well as Tosin Akinpelu from Microsoft Canada. So these are people in STEAM industries that agreed to spend their time doing this work with student projects to make sure it was the most authentic and relevant experience possible for everybody. Cool. And it led to this, the students that you're going to meet today. I'm not going to give away too much about the work that they did because that's what the rest of today's presentation is about. But it's a real nice cross-section of the projects that we received. One thing that I think is really important is that we had a middle school team that was in the top 3, that was in that final consideration for going into space. It wasn't just high school students. And in fact, we didn't have any grade 10 and 11 teams that made it. They were grade 8, grade 9, and grade 10. Those were our 3 finalist teams. I think that's amazing. And I think it's really important because it's a reminder to all of us that with the right support, we can be engaging with really complex STEAM ideas at maybe a bit of a younger age than most people think. And that's part of what we're striving for in the school district, to make sure that engagement is happening, not too young, not before it's relevant for children, but at a time when they really can start to deconstruct these complex ideas and it becomes meaningful and important for them. And I believe for these 3 student groups, this was as meaningful and powerful of an learning experiences as maybe they'll ever get in their school lives. And that's really the power of SSEP. Nicole? Now, it turns out there's a bit of a theme between the 3 finalists. You're going to see there's a kind of a real focus on astronaut well-being and ability to maintain themselves in space. It's probably not a surprise because it's kind of a really important topic. And if we can learn from that, if out of this there's any takeaway that the world learns from, it's going to help us as a global community going forward. And I think that's really powerful. But I did want to represent a little bit that actually there's a broad range of really creative ideas that came out of the project submissions that occurred. And they range from, dealing with pollutants and chemicals in the space environment, the ability of things like bugs to propagate and survive. We see mealworms here at the top. That's not necessarily just about growing mealworms, but whether or not understanding whether or not they could be a source of nutrients for human beings in space if necessary. The effect of space on things like on human parts like the eyeball, the issues related to sanitation, as well as growth of vegetation, growth of we had some fantastic projects that really could have made the cut around things like growing mushrooms and such. This array of amazing ideas, all of which would have been worth exploring. Unfortunately, we had to go through this process of getting down to a final 3 to submit. And we focus on this final 3 for a reason, because we as a school district or SFU don't make the choice about number 1. That's actually made by the SSEP organization. What we get to do locally is submit our top 3. And these are the 3 groups that you're going to learn about today. They were all forwarded as the final potential candidates, and the SSEP organization made the final decision on which one was going into space. So that's kind of the hierarchy of how it all works. Really amazing process. And the fact that every student that's on the call today got to the point where we submitted them as one that should be able to go into space is, I think, something they need to be proud of for the rest of their lives. Cool. But I also really want to point out the engagement doesn't stop there. We needed to have ways to engage on a broader sense, including at elementary. And it's not realistic in SSEP for elementary students to be preparing the type of proposals that would go forward for space consideration, but we want them engaged anyways. And also there's lots of students in grade 6 to 12, which for us is middle and high school, for them to engage, even if they don't want to do a formal proposal. So there's a couple of important STEAM related elements to it that even if you didn't do that full proposal, you were actively engaged in the SSEP project throughout the district. One of those ways is through art. The project gives us the opportunity to have 2 pieces of art, one for younger students, one for slightly older students, to be attached to the project and actually go into space themselves as a representation of the project, as a symbol of the project. These 2 patches you see here are the 2 winning patches. One was created by Daniel Pan, a grade 5 student at Lee Elementary in our district. And the second one, Grace Mosley, grade 7 at Hillcrest Middle. These 2 patches are now the official symbol of SSEP Mission 17 in the Coquitlam School District. And sorry, I said 17, I meant 16 in the Coquitlam School District. So these patches, we had hundreds of them made. They're distributed to all the students who participated in various activities. and they're the symbol of the project now, and they get to go into space with the project, which is cool in its own right. And so that's another way that we engage more students in this real science activity, but more broadly turning it just from science into true STEM, true STEAM. Nicole? And then further to this, we provided many, many lessons to teachers to engage in microgravity experiment activities here on Earth, to do little real live outdoor and indoor different experiments that are the same type of things that would lead to science experiments that may one day become proposals, SSEP proposals of their own, but where students maybe weren't ready to go down that full SSE proposal route. They could still engage in the SSEP learning, in that idea of science and its relationship to the earth, and how we practice experimentation around that. And so there were lessons occurring all over the district over that 3-month period related to SSEP that weren't requiring the students to do a full proposal. And I think that's really important too, because it's reflective of how broad the touch of this SSEP project is. It really is a district-wide activity for a period of time that focuses everybody's attention on our STEAM priority, engages kids in authentic science, and at the end of the day, we have a couple hundred who submit formal proposals, and it works its way down to these final 3 amazing projects that you're going to learn about today. So I think with that in mind, it's time for me to pass it on to Cynthia to start talking about the projects a little bit, because they're the main show.
Thank you. Thank you so much, Stephen. Quite a learning journey indeed. I will echo that. And we were always blown away by the hundreds of proposals received from elementary, middle school, and high school from the Coquitlam School District. That was amazing. So I think this morning really is a celebration of all the hard work that all the students did for this project. And this morning you will see 3 finalists, as Stephen had mentioned, 3 finalist experiment proposals that were submitted and selected for this SSEP program. So before I begin handing it over to the first group, let me just give a few reminders on house rules. We are using a webinar format for this morning over Zoom, so live transcription has been enabled. You have that option of turning that on if you find that helpful. This session is also being recorded. The link will be shared broadly after today's event. The chat page has been turned off, but you do have a Q&A box that you can access. We encourage you to type in any questions or comments you have for each of the presenting groups. Any questions that you have for the students, please feel free to type that on the Q&A box. Also, after today's event, we will be sending you by email a feedback web survey link. We encourage you to fill that out. We'd love to hear some comments or suggestions that you might have about this event. All right, so enough of all those introductory spiels. I think it's really more about spotlighting the students' finalists and to allow them to showcase their hard work that they've been working on, as Stephen had mentioned. So we'll start off with Hillcrest, second will be Charles Best, and third will be Port Moody Secondary. So let's start off with the first one. We have Marisa Bassetto and Selina Zhang, both grade 8 students from Hillcrest Middle School, and the title of their presentation is Growing Bacillus Subtilis Using Nutrient Agar in Microgravity. So let's hand it over now to Marisa and Selina. Over to you.
Our experiment is growing Bacillus subtilis using nutrient agar in microgravity.
My name is Marisa Bassetto.
And my name is Selina Zhang.
We are in grade 8. Our science teacher is Mr. Engelman from Hillcrest Middle.
Our experiment idea is sending Bacillus subtilis on nutrient agar in space. Bacillus subtilis is safe and easy to grow, and nutrient agar is a growth medium. meaning it grows microorganisms like bacteria and fungi. It grows colonies so that we can see them without a microscope. What we planned on doing is to grow bacteria using nutrient agar to see the colonies and see how microgravity would change the way it grows.
Originally, when we went to work on this project, we knew next to nothing. Everyone else in our class had at least a vague idea on what they wanted to do, but Selina and I could not agree on anything. Every single one of our ideas sounded not good enough and really boring.
Fast forward, it is now the second week and we still have no good ideas. The only ones we had were very bad. The first one was dissecting strawberry DNA, which sounds cool, but really wasn't beneficial and would definitely not work. The second idea we had was making nylon combining cyclohexane and hexamethylene diamine. However, as should have been expected, neither of the chemicals were on the list of things that could be in our experiment because the chemicals would be too risky to have in a tiny plastic tube. So that idea was quickly shot down.
Week 3 rolls by. We were sulking in our inability to come up with anything when an idea struck us that would eventually lead us to our end project, the yeast idea. This idea was to grow yeast in space. It started out very simply. This is when Selina, while researching, came across nutrient agar and had the idea of adding nutrient broth, which is basically just liquefied nutrient agar, but because yeast grows better in a liquid environment. Now, because this idea of growing yeast in nutrient broth was very common, we decided to make it a bit different by switching the nutrient broth to nutrient agar. We stuck with this idea for a while until we found that almost our exact project had been performed by NASA in space, the nutrient agar and all.
And we realized we couldn't spend very much time on the yeast idea since it was much more of a common idea than we originally thought. And our idea had to be unique in order to be selected. That was our mindset going through the process. This is when we decided to go through all of the materials on the material list and started researching bacteria that were named, since we could easily replace the yeast with bacteria and not have to start from scratch. We went through tons of ideas. We settled down on at least 7 ideas, and every single one had a huge con that would lower our chances of getting selected by a lot. We started getting discouraged after many days of indecision.
Finally, with roughly 3 weeks to spare, we got our final idea. Bacillus subtilis, otherwise known as hay bacillus or B subtilis, it was almost a perfect idea. We just needed to put some research into it. This is where puromycin dihydrochloride comes into play, an idea that we knew would be quite surprising and out of the ordinary. Puromycin works by making bacteria go docile. This was perfect since after the experiment was performed in microgravity, the bacteria would need to stop growing because if it were to continue, not all of the bacteria would be growing in space, so it would have to stop growing somehow before it got back to Earth, and the puromycin was the perfect idea. Combined with the nutrient agar slant on our idea couldn't have been performed before in the same way, which was our main concern.
Now keep in mind, we were very behind. Compared to the rest of our classmates who had tens of pages of complex mathematics theorems in their proposals, our project was sad. Lots of our classmates were finished by the time we had started working on our final proposal. Not to mention, we had no idea what we were doing most of the time, and we were really stressed about everything.
With a lot of overthinking, learning, and work, we were able to get our proposal done, and obviously well enough since we got selected. The moral of our story is to persevere and keep on trying, even if you don't know anything about your topic.
Now that you know the background of how we made this experiment and what it is, let's go more in depth. What we planned on doing is to grow bacteria using nutrient agar. On the left side, we would have nutrient agar with freeze-dried bacteria. Distilled water is in the middle. When the clamp in between those two volumes is removed, the astronaut would shake the tube and rehydrate the bacteria. After 2 days of growing, the second clamp, which is the clamp between the middle and the right volume, would also be removed.
Then the growth inhibitor, puromycin, is released. It would stop the growing and will make microgravity the only variable. We would have also been doing an experiment on Earth at the same time. This experiment is to compare our results with the other experiment on the ISS.
If our experiment went to space, this would be how we would look at the data. We would first look at any differences that we can see at first glance. Any major differences should be obvious. For example, if the colonies were growing in a weird pattern or had a different color, then we would put the two samples of the experiment under a microscope to analyze the results.
One way we wanted to analyze how the bacteria mutated was by taking two small samples of two experiments and putting them both on an agar plate to see how they would react to each other. There was just one problem with the pure meisin dihydrochloride. If the bacteria can't grow, then we can't see the mutations. We would need to dilute the growth inhibitor. Though our experiment may sound well planned, there were and still are many things wrong with our experiment that would need changing, stuff we didn't even think of. One of the problems is that even if we were going to freeze dry the bacteria, it wouldn't stop the growth. That is because agar has a lot of water in it. It would rehydrate the bacteria before it even went to space. That is a big problem because we needed microgravity to be the main variable. We don't want anything else to affect the results.
The other problem is that agar has to be sterilized. If the agar isn't sterilized, all sorts of random bacteria that we don't want could grow, and it could mess up our results a lot. One way we could sterilize it is using an autoclave. An autoclave uses steam to disinfect and kill the microorganisms. This was a problem mainly because we didn't mention where we were getting the autoclave from, and they're really expensive. We also forgot to go over the autoclave sterilization process in our proposal.
Our experiment is helpful to learn more about bacteria in general, whether they grow fast or slower in microgravity, whether they adapt quickly to the new environment, or the other things that might happen in strain conditions. Bacteria is essential for decomposing plants and recycling nutrients, and it's also very important for the human body to function. The Subtilis is the safest and easiest bacteria to work with, and it's also the most studied bacteria. If our experiment worked in microgravity, we could grow other things on nutrient agar slants in the future.
Thank you everyone for listening to our presentation.
Thank you so much, Marisa and Selina. That was awesome work, and I do appreciate the perseverance and hard work you were speaking about. So awesome work. So I'd like to take a few moments to pause and allow our attendees to type in their questions or comments they might have from Marisa and Selina. Please feel free to use the Q&A box you have on your screen and type in your questions or comments. We will get to all questions after the third presentation for this morning. All right, thank you so much. So let's move on to our second presentation for this morning. This time we have Jisong Han. She's a grade 10 student from Dr. Charles Best Secondary School, and the title of her presentation is The Effects of Microgravity on the Growth Pattern and Germination of Buckwheat. Over to you, Jisong.
Okay, so my project, well, my experimental design was on the effects of microgravity on the growth and germination of buckwheat. And yeah, so my name is Jisong Han, and I'm a grade 10, and I attend Dr. Charles Best Secondary School. And my teacher at the time when I was participating in this program was Miss Hamilton. So the reason why I chose buckwheat, a lot of people are actually curious about this because buckwheat is never a common plant. But the reason I chose it was because first I decided on testing the germination of a plant in space because I was reading a botany book and I was kind of interested in plants at that time. So I was actually having buckwheat noodles at the time. And then I just had that blink of a thought that it wouldn't be a bad idea to try to send it up to space so the astronauts can also enjoy this amazing plant. So I did some research and I found out that buckwheat is actually high in minerals and vitamins, And it also has this substance called rutin, and it has fiber, which will help with health problems related to space, yeah, such as high blood pressure. So a short description of an experiment was that buckwheat seeds will be essentially sent up to space, and water will initiate their germination process. And then their germination process will be stopped with a fixative solution because I want it to be stopped before it can reach Earth. And then the seeds would be compared to the control group that was raised on Earth. This investigation would be providing data about the germination of buckwheat in microgravity, which would contribute to the acknowledgement of potential merits that planting buckwheat offers to space exploration. And experimental design. So my question was, can buckwheat germinate under the influence of microgravity? And if so, how would it differ? So as you can see, there is two parts of the question, which is one is, can buckwheat germinate under the influence of microgravity? My hypothesis for this part of the question was that it can, because there's a lot of other experiments that was already done on the ISS by NASA that stated that a lot of other plants, such as lettuce, mustard, and even wheat, can grow in the ISS under microgravity's environment. Yeah, and for the how would it differ part of the question, my null hypothesis was that the roots would possibly be less straight, as in they'd be curled up, or they'd be facing another way than the bottom, because a plant uses gravity to figure out which way is the down way and which is the up way. And also an experiment called CARA suggested that cytokinin, which is a plant hormone that plays a role in regulating tissue growth and cell division in the roots, is affected by microgravity because the substance, the gravity is not there to pull the substance down. Yeah. And yeah, for the experiment, the experimental group was the group on Earth, which would just normally grow on the circumstances of Earth's gravity and Earth's environment. And the control group was that. And the experimental group was that it would grow in space under the microgravity and the space radiation and all that stuff. But except for that, both groups would have same conditions to grow in. And the dependent variable would essentially be the length, shape, mass, and germination rate of mainly the roots, and the amount of niacin that is involved in the buckwheat plants. Yeah, next slide, please. Okay, so observation, that's an important part. I was going to observe the length, shape, mass, and rate and amount of niacin, and the method used would be the length and the shape and things like that would be under the microscope, and colorimetric technique for the amount of niacin. And analysis, I was going to do a two-tail test to tell me if the numbers that I've gathered from the data were significant enough. And the colorimetric technique was going to be used for the, as I mentioned before, amount of niacin. Yeah, in which the amount of niacin would be extracted from the buckwheat plant, and it be reacted with another substance to create a yellow color, and the intensity of the yellow color will tell me how much niacin there is in the buckwheat plant. And the conclusion would be essentially drawn from the analysis. So why is this experiment important? Well, first, it offers potential health benefit that will help with space-related health problems, yeah, such as high blood pressure and just basically circulation problems in general that can be caused in microgravity. And it can also be grown to edible state in water, only using water, which would save the astronauts from the pain of trying to send a bunch of soil into space, which would basically take up a lot of space in that rocket. And it can provide insights to improve current scientific knowledge, as, for example, it can provide more information about how plants direct their roots, and how gravity just affects how they grow in general. Yeah, and then also basically, of course, it would provide information about if buckwheat would grow in space and if it's a good candidate to grow in space. And the difficulties, well, that's an interesting slide. First, for me, it was deciding on the question. I am not a decisive person. I'm actually indecisive. I changed the question, I swear, 5 times when I was doing this, and I had 1 month. So yeah, like at first I was gonna do something chemistry related, which is completely not really completely, but kind of different than what I'm doing right now. And finding ways to test the nutritional values in grain, there was almost no kind of research that indicates, that shows me how to find the nutritional values in grain, how to measure them. And searching for professional advisors, I emailed a lot of experts with botany, experts in botany, but I didn't really get that much replies from them. Yeah, so that was kind of hard, but I managed to do that. And verifying the reliability of sources, I think that's a hard one for a lot of people. But for me, I have this method. So I just go to a reliable scientific website, such as NASA or a government agency, I just start from there. And then I search up what I want to find from that website, which would give me reliable information. Yeah. But your digital literacy teacher would have a lot to say about this. So yeah. And that's it. Yeah, thanks for watching.
Thank you so much, Jisong. It's apparent how much research work you did, and I'm amazed by the hard work you did for this project. It would be quite awesome to germinate buckwheat in space someday. So thank you so much for your work, Jisong. So let's take a few moments at this point to allow our attendees to type in some questions or comments they have for Jisong on their Q&A box. All right, keep those questions coming. Thank you to those who are typing on the Q&A box. We appreciate your engaging with us. So we're done with Hillcrest and Best. Let's move on to Port Moody Secondary, and we have a couple of grade 9 students, Jong-Un Lee and Yong Lee, and the title of their presentation is Changes in Beneficial Metabolites by Lactic Acid Bacteria in Microgravity. J.E. and Yong, over to you.
Okay, so the changes in beneficial metabolites by lactic acid bacteria in microgravity.
We're excited to share with you our project that will be conducted on the International Space Station. It is an honor to do this project. And without further ado, let us introduce ourselves. Hello, I'm J.E.
And hello everyone, my name is Yang. And this is Miss Mahai, our science teacher. Unfortunately, she couldn't join us today, but we would like to thank her for her amazing support in this project throughout. We are all from Port Moody Secondary School. So let us tell you a bit about how we got interested in science. I got interested in science by watching a lot of science TV shows, which fascinated me in human anatomy and psychology. Science is also something that connects and helps make sense of our world, which motivates me to continue to learn how it will be innovated and developed in the future.
I got interested in science by watching YouTube videos. There's a channel called Kurzgesagt, which sparked my curiosity for science. I remember the first video was about the immune system. After that, I was hooked. Many videos later, I watched about microbes and their effects. So, are bacteria good or are they bad?
The answer is neither. What's fascinating is that bacteria do more than you think. Some are less than pleasant to live with, like salmonella, which is the same stuff that gives you food poisoning. But in contrast, some are friendly and help you live. For example, many strains of Lactobacillus, the same stuff that ferments yogurt, is not only tasty, but also produce important things such as
energy for the cells lining your gut, some vitamins, and a hormone. They also help absorb the nutrients from the food you eat.
They also produce some vitamins such as vitamins D and K. These are important vitamins for bone health. There is a reason why vitamin D is added to milk. It helps absorb calcium, which is the predominant mineral in your bones.
Vitamin K is needed to turn the calcium into bone. This is important because astronauts tend to lose bone mass and it can be expedited by a deficiency of these vitamins. Did you know that 90% of the serotonin produced in the body is made with the help of the bacteria in your gut? If you've heard about serotonin before, you may know it is a thing inside your brain that affects mood. Well, in this case, it has different effects because it is not inside the brain, but it helps the hormone that regulates sugar in the blood.
Human missions beyond the moon can take advantage of this because in space, we don't use our muscles as much as we do on Earth, and not moving tends to decrease the effectiveness of this hormone. Free energy, well, sort of. The bacteria also break down the food you cannot break down and create food for the cells lining the gut. Cells can use this food and then create energy. With this energy that would have gone to waste, they can grow and increase the surface area of the digestive tract, increasing the absorption of nutrients.
The body has an interesting rule: use it or lose it. When people experience weightlessness for extended periods, such as living in the International Space Station for 2 weeks, they lose muscle mass because the body has no incentive to keep it. A factor that may speed up muscle loss may be the lack of energy. Increasing the absorption of nutrients by the astronauts may help slow it down by increasing the energy the body has to use, so it's less wanting to burn muscle mass.
So with all these cool and important things in mind, we wonder how microgravity might affect our little companions, because if they change, this may negatively affect our future astronauts' health, such as loss of body mass. This may affect the decisions on which food the astronauts will consume in space. For example, if the bacteria stop producing the vitamins, they may need to supplement it or else be in risk of deficiencies.
We had to get a little creative for experiment. NASA has a list of things you can use in your experiment for safety. So, working with the materials, we came up with this. The food that will be fermented by the bacteria will be chia seeds, which are high in fiber and indigestible to the human body. The bacteria will be from a probiotic supplement. Then, to kill the bacteria, there would be isopropyl alcohol. This worked out well because things like chia seeds are what astronauts would actually eat for fiber, not a laboratory-grade block of wood.
So this is the tube that we will send to space. So one part of the tubes would be filled with chia seeds, the middle with bacteria, and the end with isopropyl alcohol. In our experiment, one would stay on the ground and the other one will go to the International Space Station. Then an astronaut would open the clamp containing the bacteria to start the fermentation. Then after 3 days, stop the fermentation by opening the other clamp containing the isopropyl alcohol. Then the tubes will come back to Earth for testing.
Then we would compare the levels of the things we talked about today on Earth. By using high-pressure liquid chromatography, or HPLC, which is a technique to measure how much of something there is, we can see what effects microgravity had on the bacteria. HPLC separates things by letting the material dissolved in a medium flow through a special column. Some things move faster than others, separating them. Then we can use various things, such as light, to measure how much of a thing there is.
So that is the experiment we designed. It is now time for some questions. If you have any questions, feel free to type them in the Q&A section. And that's our time today. Thank you for watching.
Thank you.
All right. Thank you so much, Jay and Yong. I'm happy to hear that you covered health of astronauts in your study. So that was quite interesting and very helpful. So thank you so much for sharing your experiment. So let's take some brief moments to allow our attendees to type their questions and comments on the Q&A box. Quite impressive experiments we have listened to this morning, and I'm sure you folks have your own questions and comments for our students who presented today. All right, so at this point, I'm going to go through the questions, and there are some questions that are addressed to each particular student group and some more generic questions addressed to all three. So I'll go down the line and I will call on the students whose questions are being addressed to. So let's begin with Marisa and Selina. There's a question for you. What makes B. subtilis a safe bacterium to grow, you think? What makes B. subtilis a safe bacterium to grow for Marisa and Selina? Any thoughts on this?
My memory's pretty bad. Sorry.
That's okay. Or is this something that you'd like to read on further, or what have your thought process been like as you were thinking through this choice?
I'm pretty sure it's safe to have in like any environment. It's just like generically safe because it's just a generic bacteria. There's nothing that's specific about it that makes it dangerous to people.
It's not dangerous or harmful for people.
Mm-hmm.
So there's nothing, there's nothing special about it that makes it harmful to people. It's just very, I don't know, normal. So it's a commonly studied bacteria because it's so generic, it can relate to a lot of other bacterias.
Right. Okay, thank you. So let's move on. We have a question for Jisong. Previous research has found that wheat grows even better in space than on Earth. Why do you think wheat grows better in space?
Oh yeah, I actually read that research. I think it was called Pesto. Yeah.
Mm-hmm.
And I believe that the data collected from that experiment stated that wheat germinates 10%. The final height of the wheat grown in microgravity was 10% higher. Yeah. Mm-hmm. I'm honestly not an expert at that area, but my assumption would be that maybe since plants have less gravity pulling it downwards, they can use more energy. As in, you can use more energy that would be normally, there's like no things pressing it down. So I believe you can use that energy to grow more upwards. But again, that's my assumption, so don't quote me on that. Yeah.
Yep. All right. Thank you. Next question is for J.E. and Yong. And the question is, why did you choose chia seeds? What were other options you were considering? Interesting point.
We chose chia seeds because that is what astronauts or people would actually eat for fiber. Because fiber is what bacteria ferment, usually inside the gut. So instead of using something that people would not eat, like, I don't know. Is it? I don't know if people eat agar, but we decided to use something that is more real world.
Right. Okay, thank you. The next question is a generic question, any one of the three groups can answer this next question, and I find it very relevant after what we've all been through. Did COVID impact your experiment proposal in any way? What were your experiences completing this project through our pandemic? Anyone want to share their thoughts or their experiences while working on this experiment proposal, through lockdowns or through all safety restrictions we had in place? What were some difficulties or challenges maybe throughout that time? Did you find it difficult to meet up, to source your materials, to get some mentors to help you?
Okay, so we did have one thing that was affected by COVID-19.
Yeah.
Mhm.
So when we were going to get help, like
For materials and stuff from people at SFU.
we had to like go on a Zoom call, and we couldn't actually meet up in person to like ask for help or anything like that. And we couldn't
Mhm.
actually do an experiment because of COVID. So we couldn't test out our experiment beforehand.
On Earth.
Yeah.
Right, right. So limitations as far as virtual meetups are concerned, and unable to go to the lab and work out your experiment in person. Quite valid. Anyone else? JE or Yong or Jisong? How did COVID impact your experiment proposal?
I agree with Marisa and Selina. Honestly, we were meeting up with experts via Zoom or a video call or a phone call. And I get nervous when I'm in a Zoom meeting for some reason. So
Yeah.
I couldn't ask them what I wanted to do. And I feel like it would have been better if I met them in person and if we were face to face with each other, and I would have discussed them about my ideas and things like that. So I feel like it would have been
Right.
better that way. But again, COVID protocols, we couldn't.
Right,
Yeah.
right. We all had to get used to that virtual platform, right? So yes, thank you for that, Jisong. Anyone else before I move on to the next question? Okay, our next question is for Jay and Yong actually. And the question is, why is gut health important for astronauts?
What do you say? Okay. Gut health is important because it is where most of the nutrients the astronauts eat will be absorbed through, and if they don't absorb it, that'll create a lot of problems. As you saw in the slideshow, there are many things the bacteria do and many things the guts does. So if that doesn't function exactly as it does on Earth, it might affect how the astronauts will live in space.
All right, thank you so much, Jong-un. Now let's move on to Jisong. The next question is for you, Jisong. Would you expect any differences with the potential use of sterile hydroponic solution versus natural soil with microbes?
Oh, you mean growing it on water instead of soil? Is that the question? The question stated?
Yeah.
Honestly, I think that I'm talking a little bit, when I'm talking about growing buckwheat into edible stay with water, I'm talking about sprouting them, so eating them as sprouts instead of a full-grown plant as the seed, like the seed itself. So I think you would eat it in a different way because you'd be eating a sprout instead of a seed. But if you're talking about fully growing them in water, I think aquaponics would kind of work for it, but I'm not sure about this topic. Yeah.
Right. Further research and trials, I guess. All right, let's move now to Marisa and Selina Hillcrest. Next question is for you, girls. Did you consider other options instead of nutrient agar for a growth platform?
We did One of them was nutrient broth, which is like a liquid version of nutrient agar, so it would grow in like,
Kinda. It's just, it helps the bacteria grow in like a liquid environment, but it, for like bacteria growing, I felt like it just would be better if we did it on a nutrient agar slant.
yeah, because you can't see it as well.
Because like
nutrient broth.
in studies and stuff, they're like usually people grow bacteria on like an agar plate,
Yeah.
and an agar slant was pretty similar to that, and that would work in a tube rather than like broth, and then the puromycin would also have to, and like the puromycin makes the bacteria go docile, and if you were to freeze dry the bacteria as well, if it were in a nutrient broth, that would kind of make it rehydrate and be alive again, which would mess up all of our results. And yeah, I think that was pretty much it. But we did consider it for a while.
Right. Well, thank you. Thank you for that. The next question is something for any one of the three student groups. And I noticed all of you included a slide on difficulties or challenges. So the question is, what helped you keep going when you were faced with these challenges? Any sources of inspiration, a little push from here and there? What helped you go through all those challenges?
Well, for us, it was a big learning experience because, well, it was a big learning experience and we were really invested into it. So if something went wrong, we didn't really want to quit either. And we're also really interested in this. So if something doesn't work, we had to figure out what didn't work and fix that.
Thank you. That's very inspiring. Anyone else? Jisong or Marisa and Selina?
For me, well, first, this opportunity was I really enjoyed this opportunity, so I just wanted to keep going, and it was really fun for me to overcome the difficulties and all that. But another reason would be that for me, when there's a difficulty or an obstacle, I have to get through it. Like there's this feeling inside me that kind of tells me, hey, you have to get through this, just do it. Yeah.
Yes.
So that was one of the things fueling me up.
I'm happy to hear that, especially when you're just itching for more answers and you're asking more questions and you just want to solve it all. So that's great to hear. So keep going. All right. Let's move on out. Sorry. Did you want to say something? Go ahead.
So when we were discouraged, we felt more like it was like a challenge and we just had to overcome it. So it wasn't like,
yeah, and pretty much whenever Selina was like kind of being like, oh, we can't do this, I was like, no, Selina, yes, we can. And whenever I was like, oh no, we can't do this, Selina was like, yes, we can. It never really had happened when we were both like, oh, we can't do this. So in the end, we kind of just like kept getting like helping each other up, and we just stayed motivated that way.
Yes. So I'm glad you were each other's cheerleader. So that's great to hear. So let's move back to Jisong. Next question is for you, Jisong. What other foods might be useful to future space colonies, you think, and why?
Other foods?
Yes.
That's an interesting question.
If it wasn't buckwheat or anything else, what other foods would you try to experiment on for the next time, maybe?
I would actually try to experiment on mugwort because it's one of my favorite foods, believe it or not. And also, it also has, I don't remember it for this moment, but it also has a lot of new values that would basically contribute to an astronaut's health. And also, mugwort is really easy to grow. It's actually kind of like a weed, as in it grows everywhere. It reproduces everywhere. It vegetative reproduces, yeah. And it doesn't take up that much space. So I think I would try that.
Mhm. Thank you, Jisong. The next question is quite interesting, and I'll start with JE and Yong for this. What does being a finalist mean to you? Let's start with JE and Yong, and we'll move on to the other two groups.
For me, it means a lot. I've been interested in science for a long time, and watching like videos about the ISS and like the cool things that they do there. And for our group's project to actually go to space was amazing. It's like a dream come true.
Yeah, we just applied for fun and we did not even think that it's going to get in the top 3. Yeah.
Well, I'm glad to hear that. What about for you, Marisa and Selina? What does it mean to be a finalist for you?
It was really exciting. We were not expecting it since we didn't think our project was that good, but when we got it, when our teacher told us the news, it was kind of a big deal because
we were celebrating and stuff.
Yeah, because it was really cool since we both really like science, and that's why we did it rather than like another option we had. But it was just awesome.
Oh, most definitely. Big reasons to celebrate. I will agree to that. Jisong, anything you'd like to add? What does being a finalist mean to you, Jisong? Is Jisong still here? Nope. All right, let's move on to the next question. I'll probably end with this question. And I'll address this. I'd like for each of the 3 groups to answer this. Is any advice for students wanting to submit proposals, experiment proposals, if we were to do this SSEP again? So tips for the next bunch of students submitting experiment proposals. Anything to share? Let's start with JE and Yong.
Okay, Jay, do you want to share first? I think I want to think about it a bit.
Okay. For us, I think it is to think about what the uses of this so what applications your project will have in everything, well, in places other than just answering the question.
Mm-hmm. Nicely said. Applications, relevance. Agree to that. What about Marisa and Selina? Any tips to share for the next batch of students doing experiment proposals, such as what you had done?
I would say to look at every detail and like look over your question, because there are a lot of like things that you have to talk about, like the details and
consider. There's a lot of things you have to consider when you're like thinking for like science experiments, because like even the pyramicin is something that people wouldn't think of, or just everything. You kind of have to be very careful. And also one of our downfalls is like not like explaining the autoclave sterilization process, which was something we didn't even think of. So I'd say make sure to think of everything.
Right.
A unique idea is also really important, because if some of your idea is already done, and it wouldn't seem as interesting. Because looking at all the experiments, I saw a lot of repetition in their experiments before. So having a unique idea is good.
Right. Agree to that. And just staying open to other possibilities and asking more questions. Nicely said. Jisong, did you have anything to add?
Oh, sorry, I couldn't
There you
unmute
go.
myself. Advice I would have is really go for the thing that you want to be interested in. So what you're interested in, as in if you're interested in biology, don't go for physics or chemistry, because it will actually make the whole experimental design process really interesting for you, because it's what you're interested in, and it's fun to find out more information about it, and conducting an actual experiment to find out new information about it too. You're contributing to that bank of knowledge at that. So I think that'd be the one advice I'd have for future SSCP participants.
Right. Oh, it's so nice to hear you say that. So I'm quite impressed by all the things that you guys shared. Thank you so much for that. I know I had said we will end with that question, but there's this last minute question that came into our Q&A box, and I can't help but read it out. It says, "This is an incredible achievement. Congratulations on all our student finalists." I can't say that enough. But this last question I wanted to end this with: what science problem or question would you like to research or investigate next after this? So let's move forward. If you were to do this experiment proposal all over again, any new science question you might want to tackle for the next time? Other new ideas maybe you might have, or new questions that are boiling in your head? Anyone in the group?
Well, what I want to do, well, I guess there are a lot of things that I wanted to do, but the first one didn't work out. So there's second one. I want to see how plants react, like how the epigenetics is a thing where you have the gene, but then there's some parts where it is inactivated so that it has different effects, sort of. So your hand is not your eye. So I wanted to see what microgravity would do with the epigenetics of a plant.
Nice. Marisa and Selina, what about you? Any new questions boiling in your head at this time?
Okay, so I specifically really was, like I said, like in our presentation, I really wanted to make nylon, which
the dangerous chemicals weren't allowed though, so that's why we didn't do it. But that's something we were really interested in.
yeah, because I think it was hexamethamine. Actually, both of them are not allowed because they both need to be in, I think, glass containers, because I think they melt through other things. I'm not 100% sure, but they're definitely not safe. But I would be really interested to see how making nylon in space would like work.
And also we also really wanted to do something related to cancer cells and stem cells as well.
Nice. Nice. Well, thank you for that. And let's move on to Jisong. Any questions that you might want to consider doing if you were to do this experiment again?
For me, it wasn't a question, but broadly a subject that I wanted to tackle before.
Sure.
First, I started with the idea of I wanted to study protein crystals, because microgravity is such an amazing area to make condition to make it protein crystals in. But that didn't work out because I couldn't find enough information on it. And another thing is that I wanted to test the effects of microgravity and space environment on the, and it came, and then we grew it again on Earth. But then the difference between the experimental group and the control group was that the experimental group had much more nutrients and it was more tasty and those things like that. So I was kind of intrigued by that, but that didn't work out because I could find no way to test out the nutrients. Well,
thank you. Thank you so much. Thank you, Jisong. It's always nice to end a session with not just answers, but more questions. So thank you all for sharing your ideas and how you think this experiment can actually move forward with more science contributions. So I appreciate your staying with us this morning and contributing to this session. And before we come to a close, I'd like to turn the floor over back to Stephen for some closing remarks for the students who presented today and for the rest of the School District 43 and anyone else joining us on this call today. Stephen?
Thank you very much, Cynthia. You know, I sit here looking at the questions come in and trying to, like, I know I couldn't answer half of them as they came in. They're so sophisticated, and to have these young people have sophisticated questions like that thrown at them and have to make sense of that is really an amazing testament to the learning that they've done and the fact that they have been actively engaged in authentic science, and they're thinking about these things at a very, very high level. And that to me is what this project is entirely about. It's about engaging kids in authentic science, going deep on a single problem, and in doing so, actually gaining some understanding of the world that you didn't have before, as well as practicing that scientific process, which is going to help you going forward forever as you look at the world around you. And that's why projects like this are so important for us to continue to do. And we look forward to working with our partners in the community and especially at SFU around making these projects a regular feature, an annual feature of activity in School District 43, and ensuring that STEAM education remains at a centerpiece in the educational experiences for all students in our district. Thank you very much to SFU. I want to thank all the students who so amazingly represent themselves and their learning. It's just so, you know, I'm so proud as a district leader to be seeing that level of achievement. You guys all make me proud, and I know you do that for your teachers too. Like, they're super proud of you. So well done. I can't imagine how your parents feel. I haven't been fortunate enough to talk to them yet. And I want to thank Nicole Cruz, who's on behind the scenes right now and is critical to making this project work on the school district side, and as well Heather Daly, who couldn't be with us today, but has done so much work pulling this all together. So thanks to all the students and our team at School District 43, and thank you to SFU for being our partner and making this happen.
Oh, it's our pleasure, Stephen, and thank you so much for allowing us to partner with you on this. And I look forward to more STEAM projects together moving forward. And I'd also like to thank Marisa and Selina from Hillcrest. We have Jisong from Charles Best, and of course, Jay and Yong from Port Moody Secondary. And also to Nicole Cruz, fabulous help you did with the slides and for helping the students with all their presentations. That was awesome work. And of course, to Stephen for joining me on this Zoom webinar this morning. Thank you so much for helping us celebrate science with us today at Science Rendezvous. Our next Science Rendezvous event, save the date, still a year ahead, May 13th, 2023. If you have any questions about this event or any suggestions that you might want to share, please don't hesitate to email sfuscienceoutreach@sfu.ca. Kind reminder, we will be sending a feedback web survey link by email to all those who joined us this morning. We look forward to hearing from you. Again, till the next Science Rendezvous event and till the next partnership with SD43, thank you so much. Take care and have a great rest of your day. Bye for now. Thank you.